Hello everyone, I am Vekutolu Rhesu, Assistant Professor from the Department of MLT at some Downtown University. Today I'll be explaining on TPHA test. So what is this TPHA test? This treponema pallidum hemagglutination, commonly known as TPHA, is a diagnostic test used to detect the dissolved amount of antibodies in the serum sample of a patient against the causative agent of syphilis. So in order to perform this TPHA test, we are going to require the TPHA kit and the specimen, that is serum. Now today I'll be explaining on the qualitative screening test. So to perform this test, firstly 4 wells are required for each test, that is the negative control, the positive control, test 1, test 2 and test 3. Now what you have to do is you have to transfer 25 microliter diluent to well 1, 3 and 4 of test. Then the next procedure is you have to transfer 100 microliter of diluent to row 2 of test. Then again you have to transfer 25 microliter serum into well 1 and mix well. Now after mixing, you have to transfer 25 microliter from well 1 to well 2. Then again mix and transfer 25 microliter from well 2 to well 3. Then again you have to mix well and discard 25 microliter from the well 3, where we have to again mix and transfer 25 microliter from well 2 to well 4. Now you have to mix well and then discard 25 microliter from the well 4. We have to repeat this same steps for the other sample, that is T2 and T3, that is test 2 and test 3. Now again you have to transfer 25 microliter of positive control in well 3 and 4. Now resuspend the test cells and the control cells which is available in the kit. Then you have to use the red dropper for the test cells and the white dropper for the control cells. So this red dropper and white dropper comes in the kit. Now you have to add 1 drop, that is 75 microliter of control cells to well 3. Then again transfer 1 drop, that is 75 microliter according to the dropper, test cells to the cell 4. Then you have to tap the plate gently, then cover the plate and incubate at room temperature for 2 hours, or you can even leave it for overnight and take the reading on the next day. So the interpretation here, the positive test, in the positive test the agglutination appears as met, whereas the negative test appear as the button at the bottom. So there should be agglutination in well 4, that is the test cells of positive control, and none in case of the negative control. Thus in this case, which I have given the image and the interpretation, the test sample 1 and 3 are positive. Positive as the agglutination appear as met. We also have the quantitative test, which is performed on the sample that is tested positive by this qualitative test. So this is all about the TPHA test.